First of all, thank you everyone for joining this panel for cell and gene therapy. I will introduce the moderator and he will take away the conversation. I just want to give a big thank you to our panelists for joining us from all over the globe. So Fabio D'Agostino from Claris Ventures, I will let you introduce everyone else. Thank you.
Thank you, Karen. Thank you for inviting me to this conference, to this meeting. I'm very pleased to be here. I'm a venture partner at Claris Ventures, which is a new VC fund based in Turin. However, I'm based in the UK, and we invest just in biotech and mainly in Europe. I'm not going to take too much time describing myself. I'm going to let the panel and all the people in this panel talk more about the investment strategy. So I'll start like anti-clockwise. So Megan, Rao, let's say Zoom is yours.
Okay, thank you so much. And thank you to the organizers for the opportunity to be on this panel. So my name is Megan Arau, and I represent Johnson & Johnson Innovation. And this was established with the sole understanding that, you know, while we have good ideas internally at J&J, we certainly rely on the external community to accelerate innovation, especially in this space. But we do you know, source for all of our sectors, which include pharma, med device, and consumer. And where I work in my role in the new venture transactions, we focus more on early stage partnerships at the Innovation Center. And I particularly help with the early evaluation as well as seeing these opportunities through our diligence and transactions process. So looking forward to today's discussion.
Thank you, Megana. So now next is Alicia. Okay, I'll let you pronounce the surname because I'm sure I got it wrong.
It wasn't enough. Yes, thank you, Fabio. Pleasure meeting you all. I'm based on the East Coast in New York, representing UCB Ventures, the corporate venture fund of UCB Pharma, the Belgium-rooted biopharma company. The fund was established in 2017 with the ambition to do equity investments in areas that are beyond the current capabilities of the mother company. And as such, cell and gene therapy was, one of the key area that our fund looked and is still looking into for investments. And our sweet spot are seed and Series A.
Thank you, Alicia. Florence Rosen, your turn.
Hello, it's a pleasure to be here. I'm a Senior Director of Investments at Fonds de Solidarité FTQ. We're based in Montreal, Quebec. We are a labor-sponsored fund with roughly $16 billion under management. We have 700,000 shareholders that can buy the fund as a retirement vehicle. We're in all sectors of the economy, but I'm part of the life science group where we invest in biotech, life science companies with a focus on therapeutics. We spend roughly 100 million every year, 50 million in direct investments in biotech companies and 50 million in specialized life science funds. We have currently 7 active life science funds, and I'm involved in the due diligence. I'm responsible for the due diligence of our portfolio of 20 companies, indication agnostic, stage agnostic. I'm also responsible for all the investment, incoming investment opportunities. And so it's a pleasure to meet you all.
Thank you, Florence. So Wei Yong Sun from Daiichi Sankyo.
Thank you, Fabio, and very nice meeting you, everyone. My name is Wei Yong Sun. I'm Senior Director, Search Evaluation of Daiichi Sankyo Business Development Group. Daiichi Sankyo is one of the largest pharmaceutical companies in Japan. We also have very strong operation both in the United States as well as in Europe. And the cell and gene therapy is an area that we have strong interest. My responsibility is to fund the research collaboration opportunity as well as the in-licensing opportunity for my company, and I have done a couple of deals in this area in the past years. Thank you.
Thank you, Leon. So now Michel Zohar, OrbiMed.
Yes, thank you, Fabio. So I'm excited to be here with all of this very professional panel. I'm part of OrbiMed. OrbiMed is one of the biggest software VC in the world, over $18 billion asset under management. I'm part of the Israeli team. We're based in Tel Aviv, responsible investment in Israel, of course, but also Europe and sometimes US, together with our colleagues in Manhattan. In OrbiMed, as I mentioned, we focus only on healthcare. I would say that 80% of our portfolio is biotech, but we do have investment in digital health and medical devices. We are stage agnostic and we are ticket agnostic, meaning that we truly looking for a strong scientific rationale before investing in a company. As for my day-to-day job, I'm responsible for the deal flow here in Israel and in Europe and managing some of our portfolio company. And I'm very excited for our discussion. So thank you, Fabio.
Thank you, Michel. Thank you all for introducing yourself. Certainly, there are so many questions, and I'm sure people attending will have many more, but maybe you can just start by discussing what's really different between investing in a cell therapy company, cell and gene therapy company, compared to traditional small molecules or biologic companies, strictly speaking.
Maybe I can start. Oh, go ahead.
Oh, okay. Okay, well, I would say, okay, small molecules, I would say they're the cute small puppy. Cell gene therapy are the big hairy monster. So we know how to handle the small puppies. We know in small molecules, I could say we know what we don't know. Cell and gene therapy, we don't always know what we don't know. Yeah. I would say in cell and gene therapy, the process is the product, whereas in small molecules, the product is the product. So they're in addition to the many manufacturing and the difficulties there's regulatory issues as well. There's only been 2 products approved by FDA, whereas in cell and gene therapy, small molecules, it's countless. I don't even know the number. Just as an example, I read something very interesting lately that this application for Spark Therapeutics was 60,000 pages for approval, and the majority was CMC. So that just tells you right there how the regulatory world is different there than for small molecules.
Thank you, Florence, and I like the example, so very good. I'm gonna borrow, I think. But thank you. Wei-Yong, you wanted to say something?
I totally agree with what Florence said. So CMC part, I think, is the most important difference between cell and gene therapy versus small molecule, because the CMC part of the cell therapy and gene therapy are just so complicated, right? So when some of the company approach me talking about their gene therapy and the cell therapy products, my first question is, how do you make it, right? So and also if the cost of goods is too high, typically it's very difficult for us to just make any profit from this kind of product, unless it is a very ultra rare disease. Like some company can charge $2 million for like one drug, but that kind of story is very, very difficult to repeat.
And I completely agree with Florence and Wei-Yong. I mean, in terms of the complexity with cell therapies, it's off the scale in comparison to small molecule deals, and particularly when all the components related to IP or just components in general that have to work together fairly seamlessly to get to the end product is something that sticks out with us, and we pay extra attention to as we evaluate opportunities.
Thank you, Miguel.
Kind of echoing what everybody said. I mean, obviously the cost of goods sold is going to be a really big issue, but also the safety and the regulatory are kind of uncharted territory in a way, meaning you have to encounter this risk, and it's hard to mitigate. kind of following someone else's footstep. Another issue that we see as crucial is the team, where in small molecule, you usually get a well-experienced team who've done this before, did few exits, sell to a big pharma. Unfortunately, in cell and gene therapy, it's still early. So you don't have a team with a lot of experience. I mean, they do have experience for academia, but not from biotech industry. So you have to really rely on the ability to execute of the team.
Yeah, if I can continue on that, the cell and gene therapy are extremely capital intensive, and the competition for the couple key talents that have brought some of the first products to the clinic is very thin. So it is a small and highly competitive ecosystem, but highly rewarding as well. So yes, and the CMC is kind of captured into big arrays and more capital intensive operations as well. So that's what we've got, what we are dealing with. Compared to the small molecules.
Yeah, it was true. I mean, I agree. I mean, with everything the fellow panelists just said, and I would say, on a positive note, what we can achieve with cell and gene therapy is sort of unprecedented, often in terms of efficacy. This is not to say that we should disregard all the other therapeutics. But like there are both, if you look at gene therapy, what is possible with gene therapy is not possible with other therapeutic areas. And that's what I've seen from many people and many VCs, at least in Europe, that have been investing in cell and gene therapy. That's what motivated them to invest in more in cell and gene therapy products. I would say that while we are talking about cost of goods and CMC, another natural follow-on question would be, we have cell therapy, cell and gene therapy, the medicinal products, we have CDMOs that actually manufacture those products, and we have all the manufacturing tools that are needed to produce this product, manufacturing hopefully at a cost which is reasonable. And certainly in terms of CDMO, there is more and more need for manufacturing capacity. Do you think that we will also need more investors or more VCs focused on manufacturing tools for these therapies?
I can start again.
Yeah. Please.
When you say manufacturing tools, what comes to mind are vector engineering companies, and they're in the news all the time now about, and the ones that just come to mind are Dino and 4D molecular and Stride and Shape, and so they're raising a lot of money. And what they're doing, they're making AAVs that are safer, that are redosable, tissue specific, larger, and ultimately easier to manufacture. And each one has their own little, little twist of those companies. So I think yes, that's what's happening. Yes. That's what's happening in the industry now.
And as you may know, UCB Ventures is one of the investor in Stride Bio, and having both the AAV platform, but also the manufacturing capabilities are it's beyond the people are two key components to have, and it's a strategic advantage to have your manufacturing capabilities in-house. It requires some efforts, but definitely worth it. And then at the other side of the spectrum, you've got other companies. I think it Elevate that raised a huge amount yesterday, 3 digits, and that's purely manufacturing. So you can also just make a case of, you're just developing a new CDMO, so definitely a business model there.
And just to add, in terms of J&J's perspective, or Janssen, specifically around oncology and cell therapies, we're looking for those, transformative approaches for large-scale and cost-effective AAV production, as well as, bringing down the cost of goods in the manufacturing process. So those are definitely areas for us to look out when we look for external opportunities.
Regarding the AAV manufacturer, the most commonly used cell line is the HEC-293 suspension, and that cell line needs to use the triple transfection, to generate AAV. And the GMP level plasmid is very, very expensive. And also it is relatively easier to scale up to 50 liter, 200 liter, but anything beyond that is very, very difficult and challenging. So that is also the reason that last year, Daiichi Sankyo did a deal with Ultragenyx. We in-licensed their technology using the HeLa stable cell line to generate AAV, and using the HeLa stable cell line, we will avoid to use the GMP level plasmid, and also that can be easily scaled up to 2,000 liter. So that is like the one of the new technology in this field.
Thank you.
I'll just add that, Magna, if you do find this major company that deals both with delivery and manufacturing, please do share.
We're looking.
Because this is a huge value proposition.
Yep.
And kind of in another angle, I would say that basically, even in the early stage, the regulatory agency that typically asks the difference between the manufacturer that's been used during the process of the early clinical trial to the upscale manufacturer. And this seems like a small gap, but it's not a small gap at all. And if you have a platform that you can use in both cases, even for small study and then for a large scale post-market study, that would be extremely valuable for all companies dealing with cell and gene therapy, in our view.
Yeah, it's a certainly interesting topic, and I think even on the manufacturing side of the business model around CDMO, I think even that is affected by COVID, and I don't know whether, probably COVID is a topic for any kind of panel these days at conferences, but having some of these manufacturing facility having to be used for vaccines or more monoclonal antibodies for other diseases. So I think that affected and limited even more like the cell and gene therapy. But on the AAV or lentiviral vector note, let's say, what do you think that are the emerging trend if you look at cell and gene therapy altogether, and what do you think are the latest sort of most relevant setbacks, if any?
Maybe I can start. So in the cell therapy area, actually we are very interested in the allogeneic CAR T. Right, so the autologous CAR T has already been approved, but it has a lot of the logistics issues. Right? If you look at the sales of the marketed product, it is not a blockbuster. However, if there are also a couple of companies using a lot allogeneic CAR T and already in clinical stage. We would expect that this is more like a big pharma friendly business model. It is similar to other like antibody or biologic drugs, and so those are like the area that we have strong interest. In the AAV gene therapy area, as Florence already mentioned, basically how to deal with the pre-existing antibody and how to AAV to have the repeat dosing. Those are like the new technology that we have strong interest in. Because the current AAV basically is only like one-time dose, and also for the patient already have pre-existing antibody, the efficacy will be decreased. And once you dose the AAV, it also will generate antibody that you cannot, do the second dose. So those are like the unmet medical needs and also the technical hurdles that both the pharmaceutical industry and the biotech company need to work together to find the solutions.
Thank you, Ian. Anyone else?
I can go. So Fabio, you mentioned a lot of things in your question. You mentioned trends and you mentioned setbacks. So I guess I'll start with, okay, I guess I'll, pardon?
One of each if you want.
One of each, oh, okay. So I'll start with, I want to mention two important setbacks which come to mind and what we can learn from them. And one of them, it's interesting because I was on this panel last year. So I'm the only veteran of this panel last year, the last one that was live before we went into pandemic, which is the JP Morgan RESI panel. And they had asked me, what was the next big thing? And I thought it was hemophilia, and everyone thought it was because BioMarin just had submitted their BLA, and everyone thought it was a shoo-in, and that to me is the biggest setback because FDA gave them a complete response letter. BioMarin showed interim phase 3 data, and FDA wanted 2 more years of safety and efficacy data. So what we're learning from that is FDA raising the bar. There weren't really any red flags. But these are single dose treatments. And the question is, how long is lifelong? So I think the issue of sustained vector expression and long-term safety is still unknown. So that's something we're learning. I think another important setback is the Audentis that had 3 deaths. And I think that was an AAV gene therapy, and the conclusion was the dosing was very high. So again, are we pushing the limits of this delivery technology? They are allowed to continue with a lower dose, but I think at the time I had heard that it was some of the highest doses ever given to patients. So it's assumed to be safe, but that's something we need to learn as well. So so now I'll just quickly talk. I don't want to hog up the time, but I'm going to trend. What I think is I don't know if it's a trend, but I think it's the next big thing is RNA therapeutics. And I think it's still part of gene therapy because a gene makes RNA, but you're actually delivering them now in AAVs. There are incredible companies, next generation LNPs delivering RNA and AAVs delivering RNA, and even now circular RNA. Rna just is raising money. Circular RNA that's more stable than the traditional one. RNA, I think it offers precision, safety, versatility. Of course, there are still challenges in delivery, but I think that is something that really we should look out for.
Thank you, Florence. Anyone else?
I think Florence, you highlighted the big setbacks that that kept everybody worried, but also are setting what do we need to do next, and working on the dosing and precision dosing, even delivery at the cell level and specificity to reach very specific cells and avoid any toxicity by just overdosing and just trying to push your transgene everywhere. So that's very much in scope. You also see that the pool of diseases with AAV traditional replacement is pretty set, and on the investor side, I think many of us are also looking into novel technologies to go on, working on bigger genes. So non-viral vectors are getting a lot of attention at the moment. That is that scenario we're definitely looking into.
I'm very interested as well in new distribution models or new distribution system to get to the gene right where it should go, because I think, for example, in the Audentes case, the dose was very high in order to get to target. So what if we can use a way to get the right amount, not too much to be toxic, but like to get to the point where it's supposed to go. It goes back to like new technologies, but it is great. So probably nobody's thinking about lentiviral vector and the challenges we had recently. So since nobody is thinking about that, I'll mention another trend. What about iPSCs? So we've seen like, let's say, moving a positive notes of companies like Blue Rock Therapeutics, Century Therapeutics. Is it the time now to invest in iPSCs companies?
I could speak from Janssen's perspective. A few years back, we saw, Legend Biotech and their data on BCMA CAR T. Obviously, we got very excited. It's autologous CAR T. Made a deal with them, a collaboration deal, and obviously, they're, while somewhat successful, there's obviously the obvious challenges with autologous CAR T. So in parallel, that kind of led us to look for other approaches, including iPSCs, and led us to the collaboration with Fate Therapeutics. So we have a 4-target collaboration and licensing deal with them that was signed last year. So certainly this is an area for us that we keep looking out for, just ways to really novel approaches to do iPSC differentiation in those clearly defined, therapeutic immune cell subtypes. And so that's something that we keep an eye out for.
Thank you.
Yeah, I mean, OrbiMed recently did quite a large investment in Centauri. I mean, just earlier this month. We truly believe in this space for various reasons. One is that the level of uncertainty compared to any other delivery system is quite mitigated, I would say. And secondly, the potential is really mind-blowing in terms of treating multi-disease and multi-organs using only one platform. But again, we are always coming back to the platform in all like different angles. And this is not the case yet. I mean, it's not well established yet, but I definitely see in the near future that companies will come with a great platform that will enable us to treat with pluripotent stem cells in many cases, many diseases, many organs.
It really sort of enables this, just treating a small group of people to potentially, millions within an indication. So that's, quite the exciting part about the.
Yes, and many like NK cells derived from iPSCs is kind of a way to make, or at least one way to make allogeneic therapies possible. Really looking forward to see sort of the data. If this is then proved, then it will be a game changing for the industry. I think the best sort of both worlds in a way. So anything else? I mean, also like to invite the people that like the attendees to ask questions to the panel. It's not a closed panel, so quite friendly people. So then, well, certainly another question it would be around, biotech. I'm sure you guys receive lots of investment proposition, lots of pitch deck. Like perhaps an advice to the attendees and the startup that are watching or will be watching this recording. What do you see lacking in pitch decks, and what do you would you like to see improved, or let's say improved in these pitch decks?
Okay, I could I could take it.
Please, Flores. Yeah.
What we often see lacking, and what I would like to see, is the "why should I care" slide. And I already said this in our prep call, so you guys have heard me say this before, but I think it sums it up very well. We just have to see why every fund sees hundreds of opportunities a year. So we just have to know why you stick out, how you differentiate yourself from the competition. And we like to see names too. I mean, we like to be impressed by KOLs, and if they don't have management because they're young, we understand, but is there a KOL that's championing your technology? That would help too.
And if the why should we care, it can be supplemented with also some data, even on the non-conf deck. I don't know if it's cell and gene therapy just being, it's also very competitive, right? But we just end up like signing NDAs, very, just too early sometimes, and yeah, would be nice to, and these are well protected products. There is no how. I don't think there is any harm in showing your data. So I think that would be nice to put them up front.
Okay.
And we understand that some of those proposals are early, and that's fine.
Sometimes I like to believe there is always a way around, not a way around, it might be misleading talking about NDAs, but like a way to protect the confidentiality and get the funding to a point where then they can disclose even a non-confidential slide deck. I know it can be difficult very early stage to get funding, but it is true. I can certainly relate to that. So the NDA cannot be signed usually at very first call, even before I met the company for the first time. At least that's not that I haven't seen that happening very often, I have to say. Yeah. Any other feedback from the panel?
So I would recommend that entrepreneur prepare different slide decks. One for investor, the other for like the pharmaceutical companies, because we care about the different things. For the investors, actually the management team, your business plan is very, very important, right? Because they will rely on you to deliver those results. But as like a pharma partner, we care more about the scientific data. The management team and your business plan is less important than your scientific data to us. The reason is, if the scientific data is good, then we can let our scientists work with you and deliver the results together. So just try to think about the different kind of the mindset of your audience.
Yeah.
And just to add on to Wei Yong's point, coming also from a big pharma, J&J, and specifically just a side note about how our J&J Innovation Center works. We basically offer both, early stage partnerships at the Innovation Center, which is where I am in South San Francisco. We have our later stage BD group, which handles the licensing and M&A, the more traditional sort of aspects of BD. We also have our strategic venture arm, who makes the long-term investments and provide, funding for, seed round all the way up to, series A, B, C. And then we also have our incubator space at JLabs. So to the point of, understanding that, I wouldn't say you necessarily have to tailor a deck a little bit, but if you just happen to come across any one of us, we would happily, kind of point it and triage it into a different group within our Innovation Center. But specifically for us at the Innovation Center, focusing on those early stage opportunities, typically preclinical proof of concept. To Wang's point, we really like to see the science and the differentiating of how that data really sticks out. We do like to see a little bit of the business strategy, only just to get a line of sight into where this science is leading you. And that helps us kind of see where we can plug in and where we could potentially collaborate. So it's a little bit of both, but definitely just a clear emphasis on the science.
Thank you, Elena.
Yeah, so you know, kind of taking you behind the scene of how we work in terms of choosing an investment and, you know, progressing in the funnel. We basically build an investment thesis, and every company that approach OrbiMed, and I guess any VC, should keep that in mind. I mean, the story should be well crafted, and it's a story that is built with layer. With the major layer is obviously the scientific data and the scientific rationale, but also, you know, there is the layer of go-to-market strategy, of competitor landscape, and so on and so forth. And in our previous call, in our prep call, I told Florence I love this "Why should I care" slide, and I couldn't agree more. Since we see hundreds of decks every year, when you get up from the table after watching a presentation or a face-to-face meeting, you remember between 3 to 4 slides. And I truly believe that any startup company should, you know, have that in mind. What are the 3-4 key slides that I want the investor to remember once they leave the deck, once they leave the meeting? And those slides should be kind of the key go home message in terms of the investment thesis of this specific company.
Yeah, that's very important. I have to say that it's 6 of us from the, like, I think 6 different countries, different companies, corporate venture, innovation arm, and traditional VCs. I think it's, in a way, sort of it's quite representative, I would say. The fact that basically it's 6 out of 6. What we want more is the differentiation, how they differentiate from the companies, from the other companies in the competition, pretty much. So it's a 6 out of 6, I would say. So we don't have any more questions. I mean, we don't have questions from the audience. It's quite shy. So I would ask you guys to
actually. I don't know if we can make this, you know, Q&A interactive, but I'm actually curious to understand who's attending. You know, it's an online event. I see there are 25 participants. We cannot get, you know, the pulse of, you know, shaking hands with people, so we don't really know who's here. I don't know if people can say if they're coming from, you know, academia or established biotech, pharma.
Yeah, I can see. Well, it's 25 minus the 7 panelists, including Karen. So it's 18. So out of 18, who is from academia? Maybe we can count. Total silence. So I take that's a no. I recognize.
Maybe we need to.
People might be able to either enter in the chat, or I think there's like a raise hand function in the Zoom. I'm not that savvy with it, but the chat might be the easiest way.
Yeah, the chat might be the, yeah, exactly. So, yeah.
Oh, that's all too old panelists.
Yes, so we'll probably, the vast majority of the audience here is probably early stage startups, as you know, that's kind of what we're geared towards. If people want to ask questions, you can always raise your hand and I can allow you to speak, and then you can ask the question yourself as opposed to writing it in the chat. So if you want to use the raise your hand function, you can go ahead if someone has a question. Apparently they're all very shy.
Yeah.
Or it's the end of the day and they're tired.
Yeah. Well, they also got lots of interesting points and lots of question answered already, I guess. We covered many interesting points here. Well, perhaps I'll continue with one more similar bit to what I asked before, but perhaps another opportunity to talk about the sector. So if you look at the, let's say, past 5 years and the future 3 to 5 years, what would be what's the thing you are happiest about the past few years, and what would you like to see happening in the next 3 to 5 years within this industry?
Happy to. Thinking about the patient's perspective, would love. I mean, we had some really good sign of efficacy with the Yescarta in liquid tumor. I think if we could see some of the CAR T going into solid tumors, and especially the hard to treat solid tumors, non-resectable tumors, getting to patients and having high efficacies, that would be pretty wonderful.
Thank you, Alicia.
That's just sharing a perspective.
Yeah, no, I mean, I agree with that, and hopefully it doesn't sound too Pollyannish to say that the response rates in certain situations are so incredible. Figuring out, translating that to not just a small subset of patients, but to really getting that efficacy rates and durability to multiple tumor types, a large scale of patients, making it very accessible. I think from J&J's perspective, I mean, the patient's always at the top of mind, and those are the things that, it's always the framework in our back of our heads anytime we evaluate opportunities like this. When, yes, it requires us to think very optimistically, years in the future, but that's where we want to go.
I think we talk a lot about the cell therapy and AAV gene therapy. One thing we have not touched too much today is gene editing technology, right? So I would hope that in 5 years we will have the first gene editing product approved by FDA.
Oh, well, thank you, Yang. That's very interesting. I mean, yeah, we probably take it for granted, or we probably include it in gene therapy, but that's a very good thing. I can tell you that I know many companies that are like, especially in the CDMO space, that they are not sure, say, okay, should we invest more in gene therapy capability, or in 5-10 years they will all do just gene editing and they will not do gene therapy anymore, like traditional AAV, or even like the non-viral methods. Is the efficacy of the efficiency of this non-viral method is it going to improve? So that's great. I think Michelle and Florence, I'm not sure they wanted to add something to that.
Sure.
Yeah.
What I'd like to,
Oh, please, Florence.
Oh, okay, sure. What I'd like to see, I guess, in the next 3 to 5 years is the leap from rare disease gene therapy, rare and ultra rare, which we're getting good at, I guess, to more less rare disease, to more common diseases. I guess the first one we'll probably see, one of the big diseases, maybe Parkinson's, we're starting to see, but that's the leap we have to make to leave the rare disease space to really have an effect in gene therapy.
I mean, Fabio, you also, I mean, in the beginning of your question, you asked what is the things that you're happiest about, and for us, I think it's in the past 4 to 5 years, we see a booming in terms of how many companies are even targeting cell gene therapy in general as a topic. And what we would like to see is this trend kind of growing, because the conversation is shifting. More and more investors are willing to take this leap and dive into this water and investing in this company, which there is a lot of unknown, as we discussed here earlier in the panel. But there is a lot of opportunity, and of course, as Magnus says, there is a lot of option for the patient. So we are very excited about that space in general.
Yeah, it is. I mean, if we look even further, like 10 years ago, more like cell therapy was more. It was, of course, in a way before the CAR T wave, and cell therapy was more something in the lab for academic research, and it was only with the influence and the contribution of pharmaceutical companies. And now we have pretty much all the big pharmaceutical companies involved in one way or another in cell and gene therapy projects. And also more and more of this pharmaceutical and drug development know-how being transferred. And so now we consider cells, I mean, especially the cell therapy, we consider cells are active ingredient of this new molecule, of these new drugs, with everything that comes with it. So the good and bad, let's say. So I think we have one question from the audience, which I will ask the person who just wrote it to clarify. It only has a nickname here. It says, would you mind digging a bit deeper into gene therapy in hemophilia space? Like in terms of what are the preferences? What are feel free to raise your hand if you want to. Then Karen, what should we do?
Yeah, well, I mean, so you had mentioned that the failure of the companies and the challenges, or not the failures, but the delays, and maybe talking about how to move forward, what you think will come of it, and if you think maybe they're going to try moving in a slightly different direction. Maybe that'll answer the question.
Yeah.
I think it is probably a bit of it's a difficult topic. It's, I think over, the months to come, everybody is, watching the big players and how they're analyzing the data. And so it's probably a little early to comment, especially when you don't have access to all of the data. So I would say, just towards the specific companies and seeing that they're taking a cautious approach and really trying to understand as much as possible what happened. But I don't know if anybody feels comfortable commenting there in more detail.
So I would say that the most difficult thing is to predict FDA's response, right? So, but we can always think about a couple of factors may affect FDA's decision. First, in this indication, whether there is already existing therapy or not. So I think that is the the bar will be totally different. And second thing is, we also touched about the immunogenicity issue or AAV. Basically, because of the immunogenicity issue, one patient may only have one chance in the whole life to get one injection, right? So that is a kind of like the hurdle we need to deal with. The third thing is, of course, in that one injection, how long that efficacy will last. That is the answer, we needed to, think about and provide the satisfied answer to FDA to get the drug approved.
Thank you, Wei-Yong. Yeah, it is difficult. I'm quite passionate comparing, and I kind of like comparing the evolution of different drugs. Even in traditional biologics, there's been a raise in the bar, and some of the things with the glycosylation. Let's say the bar has been raised over the years. If you compare like the 80s till now, probably the same is gonna happen for cell and gene therapy, even because more and more drugs are approved. So one thing is approving a drug where there is no other option. I think that also has an impact. And when there are other options available, then probably regulators can become a bit more picky in terms of whether the drug is worth, in a way, the risk or not. Yeah, that's sort of my view in general. Hope that answer the question. Well, I wonder whether there are any other questions, or we should probably Of course, feel free to ask a question with like between each other if you have some.
Okay. It sounds like both the audience and the panelists have decided that we have addressed a lot of what is currently on everyone's mind in cell and gene therapy. So with that, I want to give a very big thank you to all of our panelists and our moderator. And if you have any more questions, I'm sure that they would be happy to answer them. You can reach out to them through the partnering platform, and you can continue any sort of conversation, or if you have any sort of specific questions regarding your company in particular. I do also want to do a more general announcement to remind everyone to look at all of the Innovators Pitch Challenge companies that are participating, and to vote for your favorites, because the companies will be getting the companies that get the most votes will be getting prizes. So just make sure to place your votes before the end of the day tomorrow. And with that, thank you very much, everyone. Have a great rest of your day and a great rest of the RESI conference.
Thank you.
Thank you, everybody.
Bye.
Thank you.
Thank you for the opportunity. Thank you.